Hello, good afternoon everyone. Today I am going to present about genetic dissection and QTL pyramiding to improve salt tolerance in rice using multiple donors. As you know, rice is a major staple food crop for more than half of the world population. Genetically, it is 430 MB rice across the tubular chromosome, and globally, rice consumption is growing continuously. And in USA, the Arkansas is the largest rice producing state, followed by California, Louisiana, Mississippi, Missouri, and the Texas. The rice production is decreasing due to salt stress. Globally, approximately 11.25 million hectares of the land is affected by salt stress. Among them, one-fifth of the irrigated land is salt-affected land, and 1.5 million hectares land is becoming unsuitable for agricultural production everywhere. In salt stage, rice is relatively tolerant in germination and grain ripening stage, but it is highly susceptible in seedling stage, panicle formation, flowering, and pollination. Seedling death occurs while the salt stage is around 10 Ishi, and the rice significantly yield reductions while the salt stage is around 4 Ishi. And you can see the both seedling and reproductive stage. The first panel, the upper panel, is without salinization in seedling and reproductive stage, and lower panel is after salt stage during seedling and the reproductive stage. You can see the clear difference between the control and the stress plants. Several breeding approaches, from the conventional breeding to the molecular breeding approaches, has been utilized by the researchers to develop the salt-tolerant rice varieties. And the marker set selections is easy to select the target QTLs, but it is challenging due to the complex genetics of the abiotic stress tolerance. Here we did the three different approaches to develop the salt-tolerant breeding lines and the salt-tolerant QTLs and the superior alleles. The approach first one is the development of the biplantal populations using the US rice variety senior and the exotic salt tolerance cultivar TCP. And we did the QTL mapping at different stages from the germination stage, seedling stage, and reproductive stage to identify the common QTL for in the whole rice growth phase. This is our screening method for seedling stage salt tolerance. 2 weeks old seedlings were exposed to salt stress. First, they were exposed to 6 EC for 3 days, and then 12 EC for 7 days in the hydroponics, and for sand culture, 18 EC for 5 days. And then we did the salt injury scoring, the visual salt injury scoring, and then other measurements like plant height, root length, chlorophyll content, sodium and potassium content. Uh, For visual salt injury score, we gave the 1 to 9 SIS scale. 1 is for the highly tolerant, 3 for the tolerance, 5 for the moderate tolerance, 7 for the susceptible, and 9 for the highly susceptible. And this figure, you can see the TCP is our donor, and senior is our recurrent parent. It's the US rice variety. In sand culture, after 18 Ishi, you can see the response of the plants here, and this is in the hydroponics condition. The healthy plant is for the donor TCP, and where the senior is the highly susceptible. And those bar graphs shows the salt injury score, shoot length, chlorophyll, dry weight, shoot sodium, and the shoot sodium potassium ratio. This is the comparison among the parents, and you can see from all the parameters, TCP is tolerant than the senior variety. This histogram shows the frequency distribution of the ILs and the parents for all the major traits, and we can clearly see that all the major traits are normally distributed and which were used for the QTL mapping study. This is the response of parents, donor TCP and recurrent parent senior in different salt concentrations, and we tested randomly some lines and the parents in different salt concentrations, and then we check the day-wise germination number of germinated seeds, and we found that 200 millimolar concentration is significantly difference in each day for the both parents. So we selected the 200 millimolar for the whole population for the germination stage screening. This figure shows the workflow showing the salt stage screening at reproductive stage, and we planted all the lines among with the parents until the booting stage, and we gave stress. You can see here the booting stage and the second panel, the second figure, and then we gave continuously ATC salt stage until the maturity stage, and we collected the reproductive stage traits related uh, measurements. This is how we did the evaluation. We placed all the three applications, stress bench and the control together, and evaluated one plants and took picture and then measure the traits. And we can see the clear difference among the parents in both control and stress conditions. And we found some of the consistent line in all replications based on the fertile panicles. The second approach is to develop the multi-planter rice populations using the two or more than two donors. Here, we use the exotic salt-tolerant donor Pukali and Hasawi, and in the US rice background Jupiter, Marmenta, and Senior. We have two populations named MPH and CFCD, and we develop the BC3 for populations using those parents. First, we did the sand culture screening, and we found the moderately salt tolerant to the tolerant breeding lines. And we selected 1818 lines from the both populations, and this figure shows the response of those selected lines in control and the two receive salt stress in hydroponics. Here you can see easily the response of the lines and the current parent and the donor parents, and we can see clearly in the both populations the selected integration lines, the ILs, the salt injury score is almost equal to the donor Pukali and Hashawi in the MPS population, and the CFCD populations, the selected lines salt injury score is around is almost near with the donor FL478, so that we can say those lines are associated phenotypically associated with the donor parents. And from the dendrogram also we can see the recurrent parents are clustered separately, uh, whereas the selected breeding lines are clustered with the donor parents like Pukali, Hasawi, and FL478. After phenotypic confirmation and the phenotypic clustering, we are interested to uh, know about the genetic difference among the lines and the parents, and then we used the 100 polymorphic SR markers, uh, and interestingly we found the selected breeding lines are genetically associated with the genetically close to the Mormenchow recurrent parent in the first population and the senior the recurrent parent in the second population, whereas the donors are clustered separately, and it suggests that those selected lines are genetically with our recurrent parent and the phenotypically they were with the donor parents. So we can say those lines are the salt tolerant lines. The third approach is the QTL pyramiding using the integration lines. For the QTL pyramiding, we use the two different approaches. The first, the pyramiding by the phenotypic selections to identify the salt tolerant lines, and the second approach is the genotypic selections from the populations involving integration lines with known QTLs, followed by the salt tolerance evaluations. And we use the salt tolerant integrations which are already identified in our studies, like the namely CLPK 757 by GN100. Those are the salt tolerance breeding lines, and the CLPK 683 and GN100. Those are the salt tolerant another lines. We use those two populations for the phenotypic selection approach, whereas we use the another population like CLPK 683 and GN520 for the genotypic selection. This is the response of the parents, like uh, in the integration lines our the main donor are the Pukali and the Nona Bukra, and the recurrent parent are Jupiter and Senior. So Pukali and the Senior cross population is the CLPK, where the Nona Bukra and Jupiter cross population is the GN population. And this is the response of the IELTS that we used for the crossing to develop the pyramidal population. And GN of 100, GN of 520, GN 683, GN of 575. This figure was taken after 5 days after 18 Ishi solstice. This is the population development approach. Uh, Here we crossed two ILs, CLPK 757 and GN100, and CLPK 683 and GN100, and develop F1, F2, and F3. We test the lines, and this is the first one is the experimental layout for the hydroponics. Second one is the 14 days old seedlings, and we gave salt stress after 14 days. And the the panel, this shows the 7 days after salt stress. And those are the parents and some of the selected lines after the salt stress. We transfer the stress plant in the soil, and some of the plant started drying, and finally we found 21 plants in one population and 41 plants in the another population. They survived, and we got seed from those stress plant, and we harvested those seeds and then again grew for the next generations. And now we harvested F4 seeds and planted for the F5 generation. And for the genotypic selection, those are the target QTL for the pyramiding from those selected ILs. Those are based on the previously QTL mapping populations. Once we fix the lines will be selected based on the visual salt injury, morphological traits, and the genotypic results. And the replicated field experiment will be conducted for the selected lines and the parental lines, and we'll keep the record for the morphological and yield attributing characters, heading date, plant height, seed set, panicle length, number of grains per panicle, grain weight, so that we can evaluate them for the superior agromorphological character. And further genetic analysis will be done to find the superior salt tolerant allele. Our plan is to do the genome resequencing of the selected lines and RNA-seq of the tolerant pyramidal lines and the parents. From all the approaches, our expected outcome is to identify the breeding lines with improved salt tolerance. Now we already identified some of the salt tolerant breeding lines, and to identify the salt tolerant genes and the superior alleles, and which will be the tools for marker seed breeding. And I would like to thank my advisor, Dr. Prashant Subudi, and my other committee members, Greenhouse Facility LSU, and the donors. This project is funded by USD and IFA. Thank you all. If you have any questions and suggestions, I'll be happy to answer them. And finally, I would like to thank the University of Florida Plant Science Symposium 2022 entire team for this opportunity. Thank you.
Well, I would like to thank Sandeep for sharing his work on salt tolerance and rice today. And even though he couldn't be here with us today, we appreciate him recording the talk and sending it to us on time. If any of you have questions regarding the research Sandeep is doing, please feel free to put it in Q&A section, and we'll make sure that we reach back to him with his questions and reach back to you with the answers.